Necessary for many of you in the audience, but I thought this might be useful to make sure that we're all on the same page. Next, I'll describe the landscape of diagnostic testing. I'll then introduce paper microfluidics and its potential for high impact and low resource settings. I'll spend a bit of time describing paper fluidic tools for automation. I'll then discuss applications of multi-step sample processing in paper and critical supporting technology. And finally, I'll close with a brief discussion of challenges and future directions. So what is point-of-care diagnostics? Well, the main goal of point-of-care diagnostics is to move testing from very high resource centralized laboratories to where the patient is located. There are many point-of-care settings that vary in terms of their resource levels. Point-of-care settings can range from the higher resource hospital ERs to clinics and physicians' offices to the lower resource settings in the developed world, such as schools and workplaces and homes, to the very lowest resource settings of the developing world. The compelling motivation for point-of-care diagnostics development is the huge worldwide disease burden. The pie chart shows the breakdown of worldwide mortality into the categories of non-communicable diseases, communicable diseases, injuries, and maternal and perinatal conditions and nutritional deficiencies. These estimates from 2008 indicate that the two largest categories of non-communicable and communicable diseases were responsible for 85% of the worldwide mortality. A more comprehensive measure of disease burden is disability-adjusted life years and includes the effects of decreased quality of life due to disease. An estimate from 2006 indicates that for a core set of infectious diseases, the disease burden is a staggering 325 million disability-adjusted life years per year. Not surprisingly, the list of high-priority infectious diseases still includes HIV/AIDS, tuberculosis, and malaria. In addition, other high-priority diseases are diarrheal diseases, respiratory diseases, and sexually transmitted diseases such as gonorrhea and chlamydia. High-priority non-communicable diseases include cardiovascular disease, cancer, and diabetes, as well as lung, gastrointestinal, and renal diseases. So how can point-of-care diagnostics help? Well, the potential positive impact of point-of-care diagnostics is multifold. Earlier interventions before the onset of clinical symptoms, avoiding misdiagnosis for conditions with common symptoms such as high fever, and access to a greater population that is not being served by the local conventional healthcare system. These can all contribute to improved health outcomes and cost savings, both on the population and individual levels. More concrete projections on the impact of the use of appropriate point-of-care diagnostics have been made. Specifically, for conditions such as malaria, TB, HIV, diarrheal diseases, and syphilis, having a diagnostic device to target a focused population with a minimum sensitivity and specificity is projected to have a significant impact in terms of disability-adjusted life years or lives saved. So the development of appropriate point-of-care diagnostic technologies could significantly reduce the burden of disease. Next, I'd like to provide some background on the current landscape of diagnostic testing with an eye towards understanding the special needs and constraints of low-resource settings. So what are the multiple characteristics that determine whether a technology is appropriate for a given application and setting? These include time to obtain the test result, the required level of training for users, requirements for instrumentation, the need for electricity, for example, for refrigeration of reagents, as well as for operation of the device, the cost of the technology, and performance. And performance includes criteria such as sensitivity, specificity, quantitative resolution, dynamic range, and ability to multiplex. So the design of an appropriate technology should target the requirements of the intended point-of-care application and setting. Current tests can be roughly categorized into three classes based on the resource level of the intended setting. There are laboratory-based tests, conventional microfluidics-based tests, and lateral flow tests. We'll take a closer look at each of these in the context of immunoassays. In the case of laboratory-based tests, these tests are composed of multi-step processes that provide high performance and are often the gold standard. For example, in the case of ELISA, or enzyme-linked immunosorbent assay, there are multiple sequential sample processing steps that are performed. These steps include sample introduction and incubation, one or more steps to label the binding event, and a step to chemically amplify the signal due to the original label for high sensitivity detection. Also key are the multiple wash steps used to minimize the nonspecific binding events in the system. So what are the trade-offs for this high performance? Well, there are multiple trade-offs. First, the assay is not rapid, taking several hours to perform the incubations, as well as requiring significant transit times for the sample to get to the lab and the results to get back to the patient and caregiver. The test requires trained personnel and specialized instrumentation, as well as electricity, for example, for refrigeration of reagent storage, The cost per test of laboratory-based testing varies depending on the specifics of the test, but can be low for high-throughput testing. Conventional microfluidics can be used to implement multi-step processes for high performance at the point of care. And I know many of you are familiar with these advantages. The small light scales of microfluidic devices can provide a rapid time to result. Automation of processes results in ease of use for minimally trained users. Microfluidics also allows for miniaturization, portability, and the instrumentation can often be battery powered and can incorporate reagents stored in dry form to remove the need for electricity. Finally, costs in this case can be competitive with the cost of laboratory testing. So conventional microfluidics has the potential to bring high performance to point-of-care settings. An example of a commercially available microfluidics-based device is the iStat from Abbott. This diagnostic platform performs testing for targets that include cardiac markers, electrolytes, and blood gases. The time to result is in minutes from a few drops of blood. Processing of samples is automated for ease of use. The system is composed of a permanent handheld instrument and a disposable cartridge, and the cost is moderate. The permanent instrument is approximately $6,500, while the cost of a single-use cartridge ranges between $7 and $40. So this is an example of a microfluidics-based diagnostic that has been successfully used in point-of-care settings such as the patient bedside in the hospital or a doctor's office. However, there are limitations to the use of conventional microfluidics-based diagnostics in point-of-care settings. First, there's a requirement for instrumentation, for pumping, and often for readout that increase the cost of the final device. Second, the complexity of microfluidic cards can lead to added costs in manufacturing. And third, commonly used materials often include the more costly glass and silicon and PDMS. All these result in a final cost of the device that's higher than can often be supported in some of the lowest resource settings. In addition to the requirement for very low cost, there are a number of other challenges that must be met for a technology to succeed in low resource settings. These include limited contact time with patients, limited training of test providers, a lack of laboratory facilities, so testing environments with uncontrolled temperatures and humidity levels, and limited local infrastructure, including a lack of cold chain for refrigeration of reagents. And finally, a lack of support for maintenance on instrumentation. And of course, the performance specifications of the application must be met. The current standard bioassay format for low resource settings is the lateral flow test. A common example, familiar to most people in the US, is the pregnancy dipstick test. It's fully disposable and low cost. It can be less than 30 cents per test. There's no need for an external pumping system. Fluid is moved through the device using capillary flow and the porous materials from which it's made. And there's also no need for instrumentation for detection of the signal. The tests are based on a simple visible signal that can be read by eye. And finally, the test is easy to use and rapid. Most tests simply require the user to apply the sample in a buffer and look for color development at the test and control lines within a period of 20 minutes. There are trade-offs for this simplicity. One trade-off is that lateral flow tests are limited to a single delivery step. So briefly, antigen in the sample flows downstream, rehydrates antibody plus label, and both travel downstream and can bind as a complex to the captured antibody at the test line. This limitation to one-step delivery translates to limited sensitivity. Additionally, lateral flow tests have a qualitative output when not used with a dedicated reader, and they're difficult to multiplex for the simultaneous detection of multiple targets. So summarizing the landscape of available diagnostics, there's this high-performance lab-based testing that requires trained personnel and facilities. Conventional microfluidics can be used to miniaturize and automate multi-step processes for many point-of-care applications, but the requirement for maintenance and higher costs make them inappropriate for the lowest resource settings. Then there are the lateral flow tests, commonly used in low resource settings, which are rapid, easy to use, and fully disposable, but are lacking in performance. So having established a need for high-performance testing appropriate for use in low-resource settings, I'd like to introduce paper microfluidics as a potential solution. Paper microfluidics is a rapidly growing subfield of microfluidics that very generally makes use of porous light paper materials. A significant advantage of the use of porous materials is that fluid moves within the microcapillary flow, so there's no need for external pumps. Another significant advantage is that these materials are lower cost than other materials that have been traditionally used. Additionally, there's a strong knowledge base to build upon in terms of work already done to develop lateral flow devices. Research in the area of paper microfluidics is very active, and to provide a thorough summary would take much more time than I have today. What I'd like to do instead is to provide some context for the rest of the discussion by highlighting two areas of focus that have the goal of enabling high-performance testing in low-resource settings. One area of focus has been enabling testing for multiple targets from a single sample. To address this, the Whitesides group has developed paper networks for multi-analyte detection. It was our article back in 2007 that really started the resurgence of interest in paper-based devices. One of their devices is shown here on the left. The device is a small branched network that enables a simultaneous detection of glucose and protein from a single urine sample. A recent highlight from the Whitesides group in this category has been the demonstration of a test that can, from a finger stick of blood, simultaneously test for 3 targets related to liver function. Additional paper-based device demonstrations in the same category include testing for other analytes that can be detected with one-step colorimetric reactions, testing for multiple similar analytes using alternate detection methods such as electrochemical detection using screen-printed electrodes on paper, and demonstrating on-device controls for calibration to increase the robustness of a colorimetric assay. Is there a question?
My question is, why, if these devices are very low cost, why would having 2 devices which can detect 2 different analytes be, wouldn't it be better than having a single device trying to detect 2 kinds of analytes at the same time?
So I think there are specific applications where you want to have, it's informative to actually have readout of 2 different things. They actually help some clinical action. And in that case, you want to actually just be able to put 1 sample down. If you have 2 separate devices, you need to actually put 2 samples down. I think also, 2 different tests, you might have to run in different ways. So I don't think we're saying that more is always better. That's not true. I think this is specifically for applications where, it's actually clinically, there's a clinical action, it's informative to have these 2 things read out together. Complementary to this, we've been working to use paper networks to perform multi-step sample processing. The idea here is that one can then translate the multi-step processes characteristic of gold standard tests like ELISA that are based in high-resource lab settings to a disposable paper-based format that's suitable for even the lowest resource settings. Our approach is to engineer paper networks to perform automated multi-step processes in a disposable. Specifically, we create paper networks with multiple inlets for each detection region. Single-step user activation then leads to automated sequential delivery of the multiple reagents to that detection region. And key to this is the ability to manipulate fluids in paper networks. And here's a demonstration of sequential delivery in a paper network. Here are different volumes of 3 colored fluids were introduced to the source pad in a single step. The image shows the staging of the fluids in the main leg of the device and for delivery of first the orange, then the yellow, and then the red fluid. An example we'll revisit later is translating the sample processing steps in ELISA to a disposable paper device. So in order to automate multi-step processes in paper, we need paper microfluidic tools to control the transport of fluids. We're developing a set of tools that serve as the analogs of pump controls and valves in conventional microfluidics. These include controls for flow rate, on switches for flow, and off switches for flow. In addition, we've been developing tools for the spatial and temporal control of patterned reagents upon rehydration. I'll briefly highlight some of these tools today. One category of flow control is usage geometry. We can learn from the well-characterized behavior of flow in conventional lateral flow strips. We know that Darcy's law for one-dimensional flow in porous media applies, where the volumetric flow rate is proportional to the pressure difference, delta P, across the column of fluid. In the case of wet outflow, shown schematically on the left, this reduces to the well-known Washburn relation, where the distance that the fluid front travels into the strip goes as a square at a time. In the case of fully wetted flow, when the fluid front is within the wicking pad, and shown schematically on the right, the velocity is approximately steady. Geometry can be used to control timing in 2 dimensions as well. As you saw in the 3-inlet structure, simple manipulation of the path length can be used to control the transport time of fluid during the wet outflow, and that follows the Washburn relation. Width can also be used with an example shown on the right. The image shows strips that contain a simple expansion at different locations downstream and a constant width strip for comparison. Flow is initially the same in all strips. Then transition to a greater width results in a deviation from the Washburn relation and a greater degree of slowing of the fluid front. So, control parameters for adjusting the transport time of the fluid front include the path length, the downstream location of a width change, and the width of the expansion or contraction. In the case of fully wetted flow, we can make use of an electrical circuit analogy to Darcy's law of flow for fluidic circuits. The pressure difference across the circuit is analogous to potential difference, the volumetric flow rate is analogous to current, and fluidic resistance depends upon the physical parameters of the system, including geometric factors describing the paper circuit. So just as in an electrical circuit, fluidic resistances in series are summed, while fluidic resistances in parallel can be added in reciprocal. So in the development of these geometric controls, it's important for us to have a way to monitor flow and measure flow rates in our paper networks. I want to briefly describe one flow visualization method that we've developed. It's an electrochemical marking method in which a high pH band is created in a low pH background of indicator dye. The schematic shows the setup for this method. Briefly, 2 wire electrodes are attached to the paper device. The positive electrode is located at the source, and the negative electrode is placed at the desired location of initial marking. For example, an initial solution of phenol red at pH 7 can be used as a yellow background solution. The dye undergoes a color change to red near pH 8, and so a reaction at the negative electrode, shown here, creates a high pH red band that can be tracked with time in the paper network. And here's an image demonstrating the electrochemical marking method. A series of red bands have been created and have propagated upwards, and the inset just shows a close-up of one of these bands. So complementary to experimentally measuring and visualizing flow is the ability to make quantitative predictions for transport in paper networks. Here, ComSol multiphysics was used to implement a simple 2D model. The model of the simple structures was first solved for the steady-state velocity field, assuming Darcy's law of flow in porous media. Then the time-varying concentration of the tracer species was calculated using the convection and diffusion module. Bringing these together, the images show a time series comparison of experimental and model results. As you can see, there's good agreement both in the shapes of the bands and the locations of the bands. The plot shows the distance versus time comparison for the experimental and model results and highlights the quantitative agreement. Thus, we have the ability to predict and control the flow rates in these simple 2D structures. Another simple demonstration of using fluidic resistances to control volumetric flow rate is shown here. In this 2-inlet Y structure, the fluidic resistance of each of the inlets can be controlled by varying the distance between the source pad and the junction of the inlets. A simple relationship holds between the ratio of these distances and the ratio of the widths of the main channel occupied by each of the fluids. The image shows that equal inlet resistances result in equal occupation of the main channel by the 2 fluids. Increasing the resistance of the yellow inlet relative to the blue inlet then results in a smaller occupation of the main channel by the yellow fluid as compared to the blue fluid. So here's a demonstration of simple pump-free control of fluids that would require 2 external pumps in conventional microfluidics. A second type of paper fluidic tool is an on-switch for fluid flow. In one example, we've been investigating the use of dissolvable sugar barriers to delay the transport of fluid within the subcircuit of the network. In the image series, the presence of a barrier in the right fork creates a small delay of the fluid front in that fork. In the lower series, a sugar barrier of greater extent was created in the right fork relative to the left fork, and results in a greater delay in the fluid front in that right fork. The density of sugar in the barrier can also be used to control the delay time. The image on the left shows a series of sugar barriers that have been dipped into solutions of varying concentrations of sucrose, allowed to dry, and then subjected to a source of dyed fluid. The plot on the right shows the distance of the fluid front in the strips versus the square root of time for the different sugar barriers. The leftmost data set is for a strip with no sugar in it. As expected, the fluid front follows the Washburn relation. For the other strips that contain sugar barriers, the fluid progression in the barrier is delayed for increasing amounts of time with increasing sugar concentration. Then after the fluid front exits the barrier region, Washburn flow resumes. The plot here demonstrates the tunability of the sugar barrier time delay as a function of the density of sugar in the barrier. As you can see, a wide range of delays is possible, from seconds to almost an hour. Coefficients of variation for the hand-dip barriers range between 11 and 24%. These are larger than we would like, and we have seen improvement in preliminary attempts to use automated methods of sugar application. An alternate on-switch or delay valve makes use of an absorbent material in contact with the porous channel. In this case, the channel is composed of nitrocellulose, and the absorbent material is cellulose. Fluid flows into the absorbent pad, producing a delay in the fluid progression in the channel compared to a control strip. The plot shows the distance of the fluid front versus the square root of time for the control strip and for strips with cellulose valves of different length. So the delay time can be increased by increasing the pad length, with coefficients of variation less than 10%. A potential advantage of this delay method over the sugar barriers delay is that there is no significant release of chemicals to downstream processes. Complementary to the on switches is an off switch that we call a dissolvable bridge. The bridges are composed of a porous matrix and pass fluid until dissolution to a permanent shutoff state. The image shows a foldable card device that includes a dissolvable bridge circuit where the bridge is composed of a sugar. The fluidics circuit is then completed when the card is closed. Fluid flows from the source pad, through the feeder material, the dissolvable bridge, and then into the delivered material. The series of images on the right shows red fluid flowing through the pathway at various times until the bridge has shut off and no longer passes fluid. The volume of fluid pass can be tuned through various parameters. The plot on the left shows the approximate volume of fluid passed by bridge circuits composed of different geometry and materials. Note that each type of bridge passes a well-defined volume of fluid before permanent shutoff, with coefficients of variation between 5 and 12%. The plot on the right shows the volume of fluid pass versus time for a representative bridge of each type. Note that bridge circuits composed of different materials have different release profiles, while changing the cross-sectional area of the bridge affects the time of flow along the same flow profile. Another method to turn off flow from multiple inlets independently is the use of pads of varying fluid capacity. The image shows a foldable card device with the source pads on one side and the paper network on the other side. The source pads of varying fluid capacity are pre-filled to saturation with water. Contact between the pads and inlets then activates the flows. The release profiles of two glass fiber pads of different fluid capacities, internicellulose inlets, are shown in the plot. The coefficient of variation of this method is approximately 10%. And here's a movie of sequential delivery using this method. A third method to control the shutoff of multiple flows independently is to use fluidic disconnects. In this method, inlet legs are submerged by varying distance into a common well, as shown schematically on the right. The level of fluid in the well drops as fluid wicks into the paper inlets, and fluid shuts off from the inlets at different times in order of shortest to longest submerged length. Thus, different volumes of multiple fluids are automatically input into the network. General controls for this volume metering method include the geometry of the fluid well, for example, the cross-sectional area of the well, and the leg geometry and composition. And so here's a movie of sequential delivery using this method. In this case, the big piece, the big cellulose piece that's running at the top, serves two functions. One is acting as a wicking pad for the sequential delivery of fluids. It's also acting to increase the fluid depletion within the well, and that gives us extra control of that fluid. The plot here shows a square of the distance of the fluid front versus time for 3 different inlets. We've characterized the reproducibility of this method and find that the coefficient of variation is between 5 and 20% in time to switch off. And finally, another category of tools that we've been developing is patterning reagents for spatial and temporal control at rehydration. For example, we can control the duration of a rehydrated pulsive reagent. In the image on the left, fluorescently labeled protein has been spotted and dried on nitrocellulose, with and without sucrose on top. Upon interaction with fluid, the reagent dried with sucrose on top dissolves in an extended pulse, compared to the spot without any sucrose. The plot on the right shows the ability to control the duration and magnitude of the rehydrated pulsive reagent by varying the amount of sucrose added. Another example in this category of tools is combining reagents that must be stored separately until use. In the image on the left, 2 spots versus 1 spot of twice the mass of reagent in each of the single spots are patterned and then subsequently rehydrated. The plot on the right shows that the combined single spots behave effectively the same as a single spot of twice the reagent mass. This ability to combine reagent depots after rehydration is useful if we want to use multi-component reagents that cannot be premixed and dried down together. So to summarize this section, we've developed paper fluidic tools that can be used to control the manipulation of fluid volumes in paper devices. The sequencing of fluids can be accomplished using source pads in folding card format, sugar barrier delays, the geometry of the inlets, and fluidic disconnects. The choice of which tool to use for a particular application will be determined by multiple factors, including the precision required and usability of the device for the intended end user. So now that you've seen some of the paper microfluidic tools that have been developed, I'd like to describe some applications using paper networks. I'd first like to briefly describe a few methods for making paper-based devices, and then I'll discuss the examples of dilution, separations, and a signal-amplified assay. The Whiteside's group first described a method for paper-based device fabrication based on channel definition using hydrophobic barriers. In that method, they use lithography to create boundaries of photoresist. Subsequently, printing wax barriers onto paper was introduced as an inexpensive alternative and has since been adopted by many groups in the field. A second class of fabrication methods is based on channel definition using physical barriers. An early example in this class of fabrication method is the use of a knife plotter to cut out paper networks. And alternatively, we've been using a CO2 laser system to cut through back porous membranes to define our networks. This method has the advantage of a rapid design and test cycle, and we can use the same instrument to fabricate the other components of our paper-based devices, such as wicking pads, source pads, and the housing, that can then be assembled into our fully disposable devices. So the first example application I'd like to discuss is sample mixing and dilution. Here's a paper network that takes in two inputs, a source and a diluent, and outputs a diluted sample. As described earlier in the talk, the lengths control the resistances of the inlets. The resistances of the inlets control the relative amounts of sample and diluent combining into that output channel. In the image on the right, increasing the length of the inlet for the diluent decreases the volumetric flow rate of the diluent into the output channel and results in a less dilute output fluid in the case of that rightmost device. The plot summarizes how the length of the dilution arm can be used to vary the concentration of the output solution. And one potential use of sample dilution is as a pretreatment step in serology assays to detect host IgM. In the second example, we've created a paper network for small molecule extraction. The input on the left is a mixture of large molecular weight component bioassay and small molecular weight component yellow dye that's simulating a complex sample. The input on the right is a clean collection buffer. The yellow dye has a higher diffusivity than the bioassay and moves across the main channel to the right. The purified yellow dye then exits the device in the region on the right of the output channel. The extract from the appropriate piece of the outlet can be recovered and assayed. And in this demonstration, an over 60-fold enrichment for the yellow dye versus the bioassay was achieved in the extract as compared to the input. Previously, the conventional microfluidic analog to this paper separations device had been used to purify a target sample, a target small molecule drug from proteins in a saliva sample. The main advantages of the paper version of the separations device is that it is pumpless, fully disposable, and low cost. Third application I'd like to describe is to incorporate chemical signal amplification into an immunoassay for high sensitivity detection. In the schematic shown here, we can use a paper network to sequentially deliver sample plus label, a rinse to remove nonspecific adsorption, and a signal amplification region, all to a common detection region. The photo shows a prototype card that we've developed that combines a simple 3-inlet geometry with a pad-based volume metering method to create an automated disposable amplified assay card. All reagents are stored dry, so the user only needs to add sample and water to the appropriate pads on the card, and then fold the card closed for activation. These are steps similar to the user steps in many commercially available conventional lateral fluidics. Detection is based on a standard sandwich immunoassay for the malaria protein PFHRP2. For the amplification, a commercially available gold enhancement reagent is used. It consists of a gold salt and a reducer, and essentially grows the original particle labels and results in a significantly darkened signal. For this demonstration, a mock sample was created by spiking a high concentration, approximately 200 nanograms per mil, of recombinant PFHRP2 into fetal bovine serum. The top image shows the original signal at 8 minutes. This is comparable to the end result in a conventional lateral flow test. The bottom image shows an amplified signal at 30 minutes, a significantly darkened signal relative to the original. So this movie is showing running of an assay. So you first saw the delivery of the original label pink, and then now the signal amplification solutions going across to turn it to a dark color. The image series at the top shows the detection region after amplification for a concentration series of the analyte. The plot below shows the concentration versus signal for the assay with rinse and amplification steps as compared to without rinse and amplification steps. The limit of detection was improved by approximately 4-fold to 4 nanograms per mil and is comparable to the limit of detection of an independently reported ELISA for PFHRP2. We've also investigated the use of the enzyme horseradish peroxidase and its substrate diaminobenzidine. In this demonstration of an ELISA on a car, both the enzyme label and substrate were stored dry. The signal for various concentrations is shown in the image series, and the dose response for the assay is shown in the plot on the right. The limit of detection was similar in this system to that for the previous gold enhancement case, so we're continuing to optimize the system for a higher gain in sensitivity. A promising aspect of this system is that we've been able to demonstrate longer-term stability of reagents stored in dry form. We first looked into dry storage of the enzyme label conjugated to antibody. The reagent was vacuum dried in glass fiber pads, and the activity assessed relative to fresh using a colorimetric assay. The data in the plot shows that a combination of trehalose and iron EDTA is critical to preserving a high level of activity relative to fresh reagents. Specifically, we were able to demonstrate approximately 80% activity after 45 degrees C storage for 5 months. Given that enzymes have the reputation for being particularly challenging to preserve well, we're pleased with this result. Similarly, for the colorimetric substrate, diaminobenzidine, we demonstrated over 90% activity under similar storage conditions. So I hope that these 3 example applications in the areas of sample, preprocessing, and signal amplification serve to show how we want to introduce additional capability using paper networks. I'd like to close this section with a brief description of some of the efforts in an area complementary to the paper microfluidics work, namely increasing point-of-care assay capability through the use of non-dedicated mobile phones. Mobile phones have the potential to improve reproducibility by limiting user errors through providing directions to users, checking process controls, or performing interpretation of results at the test line. Mobile phones also have the potential to expand readout possibilities for assays, including enabling the collection of data from more sensitive, non-visible labels, and enabling the output of quantitative assay data. Finally, mobile phones could be used for data transmission to enable communication with the clinician or to report surveillance data. And I want to give 3 brief examples from literature. The Whitesides group first demonstrated the use of a cell phone camera for direct acquisition of endpoint intensity measurements from a paper assay. Stevens and Yeager had used a smartphone to acquire kinetic data, reporting shorter assay times and a larger dynamic range than when using endpoint measurements for an enzyme-based assay system. And the OSCAN group has developed inexpensive compact adapters to be used with a non-dedicated smartphone for high-sensitivity fluorescent and dark field imaging capability, as well as for use with colorimetric conventional lateral flow tests to enable more robust readout of assay results. Though this adapter model introduces a small piece of equipment into the system, it also has some significant positives. Since the adapter can be made to fit in lock-and-key fashion to both the smartphone and the disposable test, user variability in camera positioning and variability in lighting conditions can be well controlled. So we have all this promising technology. Where do we go from here? The ongoing challenge in point-of-care diagnostics development continues to be to create tests that are appropriate for the intended application and setting. I hope that I've made a convincing case that paper-based microfluidics is especially well suited to addressing the constraints of the lowest resource settings. Coupled with recent advances in the use of mobile phones as readout devices, there's great potential for creating sophisticated assays appropriate for these settings. The main challenges in the field moving forward are to develop a set of robust and precise paper fluidic tools for automation of devices, to develop high-performance reagents and materials, and to create integrated systems for sample in to result out, including sample transfer and pre-treatment. And I want to close with a brief description of a project in which I'm a co-investigator and is led by Paul Yeager from the University of Washington that's trying to meet some of these challenges in the context of developing an influenza diagnostic test. So why the need for an influenza diagnostic? Well, influenza causes 36,000 deaths and 200,000 hospitalizations each year in the US, and results in about $10.4 billion in direct medical costs. There are actually multiple FDA-approved conventional lateral flow tests on the market. These have been criticized for having very poor sensitivity, with reports in the literature of clinical sensitivity between 11 and 71%. Further, during the flu pandemic of 2009, the Centers for Disease Control actually called for discontinuation of the use of these lateral flow tests for flu detection. So here's a clear case of a need for a higher sensitivity point-of-care test than is currently available. The initial target setting for our device in development is the physician's office or clinic. Additional potential target settings include hospital ERs during off hours when the hospital lab is closed, and military units. The potential positive results of accurate diagnosis include initiation of antiviral therapy as early as possible, isolation of infected persons to avoid outbreaks, the reduction of antibiotic use due to misdiagnosis, and the reduction of other more costly diagnostic testing for related respiratory infections. So we're using the paper network technology for this application. The core assay will differentiate between flu A and flu B using the highly conserved viral protein nucleoprotein, while a subtyping assay for the surface-bound hemagglutinin protein will be run in parallel. We have multiple team members. David Baker's lab at UW is developing synthetic high-affinity binders as an alternative to antibodies. GE Global Research is developing substrates with low flow rate variability and low nonspecific binding. PATH, a Seattle nonprofit, is providing expertise in assay development for low resource settings. Dr. England of Seattle Children's Hospital is an expert in respiratory infectious disease and is providing flu samples for test validation. My lab is making contributions in flow tools development and in optimizing the signal output from the amplified paper network assay. And the bulk of the paper network assay development, including integration, is occurring at the UW. So I've tried to emphasize throughout the talk the importance of creating diagnostic devices that are appropriate for use in the target setting and by the target end user. So I want to share an example of how we're informing our device design on the influenza project. Our partner PATH conducted a device usability session attended by clinicians, nurses, and lab personnel to gather feedback on 2 prototype device designs that featured very different volume metering methods. The participants were provided with non-functional prototype devices, shown schematically here. Prototype A is based on a folding card format with pad-based delivery of fluids that you saw earlier, while prototype B uses fluidic disconnects to meter fluid volumes. And so a key piece of feedback that we received is that the orientation dependence of prototype B is a critical disadvantage in clinical settings in which level counter space is often limited, and there's an additional concern that the device may be knocked over while it's left running. So this is a case in which the 2 methods are similar in performance, so there's really no technical reason for us to choose one over the other. But this feedback from potential end users has focused us on moving forward with prototype A. For this project, a great strength is that we have a team that has expertise and experience not only to produce an appropriate prototype design, but to potentially translate the output of the project into a product. Other potential future disease targets for higher sensitivity detection are listed here, including malaria, HIV, measles, chlamydia, and gonorrhea. And though the focus of this talk has been on the application area of disease diagnosis in humans, paper microfluidics also has great potential for other related areas that have relevance to global health, including environmental monitoring, food safety, and veterinary medicine. And a definite advantage of utilizing some of these other application areas with respect to getting products out is that in more timely manners, that would be no need for FDA approval. And so finally, I'd like to acknowledge my close collaborators, Professors Paul Yeager and Barry Lutz, members of our labs, and the funding sources that have supported this work. Thank you, and I'm happy to take questions.
For example, the prototype A that you showed, what type of volume consumption is involved as far as, let's say, the sample, target sample versus, let's say, the water or the buffers?
So in the specific demo that I showed here, it was 10 microliters of sample, 10 microliters of rinse, and 40 microliters of the signal amplification reagent. But so, I mean, we hope that these devices are configurable. So the network would just look different if you wanted to add different volumes, but that's kind of the scale. Yes?
I was noticing that most of your work was published in Chemistry Chip. I was wondering if you guys have put up these designs and protocols in an open access location such that.
That's a great question. So we actually have a website that we set up a couple of years ago, MF 2.0, Microfluidics 2.0. And so the plan had always been to get the stuff out there. We're mainly limited by time and resources, but that would be a great place to, I mean, I know that we're trying to get some of the some of the designs out for like that. There are a number of people interested in the flow visualization method. So the research scientist in our group who would actually develop that, he has the materials, I think, ready to go. I don't know if it's been posted yet. But so that's, I think that would be kind of a good place for us to be putting that stuff up. But definitely, and if you have any, definitely if you have any, if anybody has any requests for specific protocols, we'd be happy to share them, and for any of the fabrication stuff.
I have one more question.
Yeah.
So you did a stability test on your HRP. I was wondering if you've done any testing on your affinity reagents, either the capture antibody or the antigen-specific antibody, whether they could also survive the fluids. From my understanding, my reading, that's one of the major issues with lateral flow tests today.
So we've done the, so in addition to drawing down the antibody, it was actually conjugated to the detection, the enzyme label in that study, it was actually conjugated to the detection antibody.
Oh, so that wasn't just HRP enzymatic activity, it was assay performance.
Oh, I'm sorry. Okay, so that's a good question, and I think that's a very good question. I'm not sure how long we've done some longer term tests. I don't think on that scale, but I think Dean Stevens in an earlier paper actually did preservation. In that case, it was actually in a flow-through format, but he was still preserving antibody within. In that case, it was antibody and gold particle in a porous matrix, and then looking at assay output. So I need to look that up. I'm not sure on what time scale he looked at that, but I know it was. So I don't think it was as long or at elevated temperature, but it was over a longer period. But that's a good question. And we have not done any systematic studies on the capture itself, which is also. But we also, in these tests, we also have just been, for our development purposes, we actually just spot capture, and then usually we're using them for our tests like within a couple of weeks, and we haven't seen an issue with that. I think longer term, I think in the lateral flow industry, these have shelf lives, right? There are lateral flow tests that have shelf lives of a year, 2 years. And I think what they often do is deposit them with a little bit of sugar. So I guess there are protocols out there. So I feel like there's a way forward because lateral flow tests do this all the time. Yeah.
Do you have any thoughts on more viscous samples or samples that are heterogeneous like sputum, and as far as interacting with paper microfluidics, or do you kind of require something that weighs uniformly?
Yeah, so that's a really great question. So obviously, if you go to really high viscosity sample for sputum, solid chunks, you have to do something upstream. So we have not had specific experience with that. In previous projects where we've worked with saliva, depending on, so I guess it really depends on your analyte, and that's where the application really is relevant in terms of can you afford to dilute your sample a little bit. Like when in our previous project where we worked with saliva, we're actually doing therapeutic drug monitoring. So our targets were in very high concentration, so we could afford to dilute quite a bit. And so that's what we did in that case for sample preprocessing. But you're right, whether it's in paper or a conventional microfluid channel, the viscosity that high is going to affect your results, and you have to do something.
So in some of these low resource settings, ambient temperature and humidity could be very, very different from what lab conditions. My intuition says these type of systems might be very susceptible to variations in ambient temperature, especially humidity. Have you guys looked into how that affects the flow?
That's a great question, and that's something that is kind of on the list of things to do. We haven't done any controlled studies in the lab for either of those things, but that's definitely something that, like you said, is an issue. I think it's something that probably leads to some of the variability in the lateral flow tests that are currently used, right? That ambient temperature can vary by such a wide way. We're hoping that at least by enclosing them, it'll help a little bit in terms of evaporation, but that has not been quantified. What the effect is. I think there was another question back there.
I might have missed it, but with your ELISA on a card or a lot of these assays, what is the enclosing? Is it plastic? What kind of plastic is it? How do you form it? What's the techniques to?
And so actually, I should have mentioned too, some people. So I brought some devices, so feel free afterwards to grab some of these. There's some conventional lateral flow tests as well as a number of devices. So this is actually one of the housings. So this is actually just Mylar adhesive. So we used to, even before we went into paper microfluidics, we used to fabricate a lot of devices by their laminates that we would just build up. So we use a CO2 laser cutter, as I mentioned. So you can actually buy these sheets of 8.5 by 11 materials. And so you can get all sorts of varieties of mylar and then adhesive on one side or both sides. And so that's how we would, cut different layers in our channels and then just layer them up. So we actually took to, at least in our prototype devices, using the same sort of mylar adhesive to cut out our housings. And it's kind of nice because it helps us actually define where, a lot of our stuff obviously now is pick and place assembly. So we can actually define where we need to put our wicking pad, the other source pads and things like that. And so, when you pass that around, you'll notice that you can actually peel off the release layer in certain places, and that's where, you peel that off and stick these down.
So what's the gold standard for sensitivity for these lateral assays right now? 71% is nice, but not.
Yeah, so and it ranges from 11 to 71%. I think the variability, at least in those values, because these are all FDA approved tests, so that they submitted documents that said, in their FDA documents they claim like 98%, or over 90% sensitivity. And I think the discrepancy is that obviously when you're going for FDA approval, you get to choose the population that you're looking at. It's well known for influenza. Children can have 3 orders of magnitude higher viral loads than adults. So I think some of these wildly varying cases, but it's still getting at the point that children are maybe the easiest target, right? Because they have such high viral load. But if you want to, elderly are also a target for flu tests in terms of they're more susceptible to developing issues and complications. So, and their loads would be a lot lower. And so I think that's the reason for this really widespread and clinical sensitivity. It's basically what people are using as their sample, their test sample. So, but in terms of gold standard, I think, gold standard in terms of what we'd like to get is certainly, closer to what they get in laboratories, like maybe for an ELISA or something like that, which is certainly, upwards of 90%. I think that's what we'd like to consistently get.
So you think?
Jeff, go ahead.
So you think the issue is like patient variability, or is it?
Well, so just in terms of that huge range, if someone's measuring 11%, but someone who's the manufacturers of the test, obviously the FDA would not approve the test, I think 11%, and they actually at some point must have gotten 90 something. I think it's the population that they're looking at, because that goes into the clinical sensitivity, right? What those viral loads were. Also, obviously what matters too is what they use for gold standard, and I think most people consider, when you're doing those types of measurements, PCRs, a gold standard.
So when we think about like point of care lateral flow tests, obviously the pregnancy test is a pretty good example. And here, the point of care is you go to Walgreens or CVS, you buy it, and then you go to the doctor, depending on the results. I'm wondering sort of what that point of care looks like in these developing regions, and sort of what sort of changes in engineering design do you anticipate having to make to maybe accommodate a slightly different point of care?
I see. So I kind of think of also point of care and how it's intended to be used.
Sure.
So even though, so the home sometimes is not any different. Even though we have electricity there, right? You're not counting on it like when you want to do one of these home pregnancy tests. So, and, so anyways, I just want to make that clarification. But in terms of, I think the lowest resource settings that we're talking about in the developing world, I think someone brought up definitely the situation that some of these areas that high humidity levels, high temperatures, right, are something that just varying ambient conditions is one challenge that we're going to have to design to, because that's going to affect standard flow rates and things like that. In terms of usability, though, I think people here are the same as people elsewhere, and I think for these lowest resource settings where you're assuming that you don't need any training, I think we really do have to think about. I think our partners at PATH have kind of taught us that tests cannot be too simple, right? So certainly no user time steps, right? Something that you have to fold at 15 minutes, right? Things like that. So I think those are kind of the end users that we have in mind, and that's why we want to take pains with all these different fluidic tools to find ones that we can use to really have automated, tests. So the kind of steps we think are okay are, at the beginning, you might have to load something, right? And then maybe there's some activation step. But, kind of no-nos for us are after that, having to check in on something at a certain time, right? And things like that.
Do you envision that being in sort of local clinic settings?
Oh, you mean, so these paper-based devices? So I think it really depends on what the clinic looks like, right? So, and so we've seen, again, these are pictures from our partners I visited, but, in the developing world, you could have very, very different, you could have actually quite a nice mid-level resource clinic, right? That looks like one, not in a rural area here or something. You would go to another place that's called a clinic. I mean, it could be just totally, messy, dirty, really no, electricity is, 50-50, or worse. So I think, so again, it kind of depends whether you have those things available, whatever setting you're designing towards, that'll really determine, I think, what you need to do in your device, right? So, because I've talked about, so paper microfluidics I don't think is necessarily great for all settings. I mean, there are some where you might need, really, really high sensitivity, right? Or quantitative results, right? I mean, I think that's a challenge, obviously. We're designing for places where you can just get a simple visible readout, right? And then it'd be nice down the line to couple it to some sort of non-dedicated reader. But in certain applications, you need a quantitative result, right? If it's in a clinician's office in the developed world, why not have a little box that can actually automatically do the reading? I think that would be actually well suited to that environment. So it's really, I think, when you're developing any diagnostic, I think the thing to do is to really go out and find out as much as you can about that end user and that end setting, and then that's what you design to. Because I don't necessarily think one test is, certainly going to, fill all these different. This range of settings. Yeah.
Hey, maybe I missed this, but I was wondering what the chemistry of the not enzymatic, the chemical amplification that you demonstrated?
Yeah, so there were two of them. So one of them, actually, I know that. So the first one, we actually use a gold label. That's why that was the one that turned pink first. And that's just like, what pregnancy tests use, right? It's like a 40 nanometer gold label. And then what we put in after that was actually, it's a commercially available, it's called a gold enhancement solution. And so in it, it's proprietary, the one that we got commercially just because it was easy to use. But it has a gold salt and a reducer in it. So when you when you flow that by the particles that have, attached to the detection region, it'll actually grow those particles bigger, and so shifts the absorption, and it turns them, kind of dark purple black. So that was one of them. The other one was the enzymatic system HRP, and it's substrate diaminobenzidine. And in that case, you get a brown precipitate. So obviously the HRP, when that binds, we don't see anything. But then when you put the substrate through, you get a dark brown. Here's one of them. So this is the one where that's just a gold bead where it gives you the pink and then it turns a lot darker. That's gold enhancement chemistry. And then the other one was this one. So the brown is due to conversion of the substrate DAP. You get this brown precipitate. And of course HRP has many different precipitates. You use TMB, get a blue color.
So what do you think of as the future of paper-based microfluidics, and especially in the developed world?
In the developing world?
Specifically here in the States, do you think that there's a future for this to replace another?
Oh, for the developed, yeah. Well, so I think the place that it has value in the developed world is certainly in the home. I think as the FDA just approved a home HIV test, right? And so I think I think that sort of testing, that you're assuming people might have the test with them in the office or in the home. That would be a kind of a comparable setting in terms of you're not assuming any electricity or anything like that. You want it to be very simple.
Thank you.
Thank you.